The last thing I want to talk about is interpretation and debugging of the calibrated data that you can get out of a plate reader. I see one question just popped up. When we calibrate with fluorescein, which wavelength should we use? So on the measurement page, we have recommendations for the wavelengths to use for each of these. For fluorescein, the preferred, my preferred numbers are excitation at 488 nanometers and emission at 530, a bandpass filter that's centered at 530 nanometers and is 530 nanometers wide. Like I said, these numbers are up on the site. And we strongly recommend that you use the numbers that are there if you can, because that way your numbers will be directly comparable to other people who measure using those same numbers. If you excite at 488, then you can put an emission filter at 530/30, you can compare directly to anyone else who did 488, 530/30. If they excited at 470 instead, the relationship is going to be similar, but not quite the same, because even though you could scale one molecule, the way that fluorescein changes is not quite the same as the way that GFP changes, and so therefore the calibration shifts a little bit. Not necessarily huge, but it's safer to just keep it the same if we can. Should the excitation wavelength be the same as the one used for the fluorescent protein later? Yes, absolutely. You always keep your instrument settings the same between your calibration and between your experimental measurement. So if you use different excitation and emission settings for your calibrant and for your experiment, then you cannot interpret your experiment using your calibrant. Have to keep them the same. All right, I'm going to move on to the last bit. So let's talk about, now that we've calibrated some plate reader data, how can we use the fact that we calibrated it to help interpret what we're seeing? So I'm going to start by saying what we've gotten out of the calibration is not quite what we want. What we've gotten out of calibration is an equivalent number of molecules of fluorescence and an equivalent number of cells of absorbance. But those include the background of how much fluorescence is in the blank cells themselves and how much absorbance is in the blank media itself. That's included even after calibration. So to get accurate estimates of cells and fluorescence per cell, we need to subtract the background. That's very simple, fortunately. We can simply take the OD measurements, subtract the average OD from blank media, and then convert that number with our particles per OD calibration unit. Similarly, to convert from the fluorescence including cells to fluorescence without cells, we just need a sample of wild type cells to measure the fluorescence of. Then we can take the fluorescence of an experimental colony, subtract the fluorescence of a wild type colony, and convert that. The sheets that we provide with the calibration protocol will do the first one, but they don't do the second one. So once you've done those estimates, the second thing that I want you to think about is the statistics for interpreting the measurements that are there. And here we're going to go to something that may be a little bit unfamiliar, which is the idea of geometric statistics. When we look at the distribution of fluorescence on a per cell basis, so I'm dipping a little out of plate reader for a moment. But when we look at what's happening on cell by cell, we often find that we get these sort of bell curves on a logarithmic scale. This is a little bit weird. This is not what we normally expect. Here on the left here, here's some of the things out of the Voigt lab, cello repressors. But there's a reason that we get these logarithmic bell shapes rather than getting bell shapes on a normal linear range as we would typically expect in most scientific actions. And that's because gene expression can be thought about as a really complicated reaction with a whole bunch of rate terms that get multiplied. And these are the rate terms for the parts we usually don't model, not the promoter, but the process of the transcriptase assembling and binding, each reaction that adds a nucleotide, each reaction, the process of the ribosome assembling and binding, each reaction that adds an amino acid onto the protein. These sorts of things, there's a huge amount of chemical reaction going on there. And the parameters vary a bit from cell to cell. And we multiply a whole bunch of parameters like that. The same mathematical central limit theorem that says if I flip a bunch of coins, I get a bell curve, says that I should get a log normal bell curve instead. This is going to apply to us in a plate reader world, even though we can't see individual cells, by interpreting how different are our replicates. Because we're going to think about the differences of our replicates being in terms not of plus minus differences, but in terms of multiplication and division and fold. So a little bit more on geometric statistics, because they may be something unfamiliar to you. The way that you can think about geometric statistics is that they're like normal statistics, only you put it on a log scale. So you take your log 10 of your data, then you compute mean and standard deviation, and you put 10 to the something, 10 to the result. That means that rather than getting plus minus error bars, you get times divide error bars. And we think about things in terms of k-fold. This is 10-fold higher than that. Those are the right statistics to look at because we're on this log scale. Here's an example. Again, we're dipping out of plate read a little bit over to the world of flow cytometry data to look at a distribution of cell behaviors to see why geometric is the right statistics. This black is a piece of real data of per cell measurements, and the geometric bell curve fit of mean and standard deviation is in blue, and the normal arithmetic fit is in red. What I want you to notice is that the geometric fit looks like what the cells are actually doing, and the arithmetic, plus minus, normal mean, normal standard deviation has nothing to do with the actual shape of that cell distribution. It's all weird. Some of you might be thinking, what about 0? Because if I take log 10 of a 0 or a negative number, I get no number. If you have a number where your net absorbance or fluorescence is at or below 0, this may happen to you when you essentially have something that's indistinguishable from the background. What you want to interpret that as is, I can't measure this number. And in fact, it's not just 0. There's some ways above 0. Things are indistinguishable from the background. If you take the your wild type cells and you look at their fluorescence, you look at their geometric mean and standard deviation of those fluorescence, that's going to give you a range in which you know 2 standard deviations up is about the point where you cannot anymore distinguish from no fluorescence at all. So when you've got low values coming close to or below 0, Don't worry about the fact that that taking a log might put you into not a number space. You just want to interpret those low values, even the ones above 0, but below 2 standard deviations above your autofluorescence, as this measure is less than this measure. I cannot see, say, less than 470 methyl per cell, or less than 1.3 times 10 to the 6 cells in my sample. That's very important for interpreting your data, because we get a lot of spurious results from people thinking that a piece of background noise is actually a difference in signal, and saying, oh, I got a 10-fold increase in behavior, when it's really just that they were a little bit, the background noise happened to be a little bit closer to 0. One more thought in interpretation. We talk a lot about controls in science, but most of the controls that get talked about are the experimental controls. There's another set of controls that I want you to think about, which are called process controls. And experimental controls, those are the ones where you're asking, is my hypothesis true? And you want them to be as close as possible to your experimental conditions, and one control for every factor that you're studying. Process controls are the converse. They're asking, are the assumptions I'm making that my instruments are working, that my protocol isn't totally broken, etc., are my assumptions okay? You're asking, should I trust the data? And there, you want your controls to be as far as possible from the experiment conditions. This is where our calibrants live. Here's some examples of experiment versus process controls with, so say that we're studying this system where we're going to use a recombinase to try and snip out a terminator to turn on this expression. Our experimental control, there's sort of an obvious thing. If the hypothesis is that the recombinase causes this snipping to turn on GFP, we should also run one without recombinase. The process controls, on the other hand, are, for example, maybe I don't know if my cells actually have the GFP in them in the first place. Did I transform them right? I might want to have a constitutive marker there to make sure my system is actually in the cells. I need to have positive controls, so I see how the fluorescence I'm getting out of the cells compares to just fluorescence at all. I need my calibrants, I need my wild type cells to compare my zeros with, etc. All these pieces to support our units. And those are all going to help us interpret when something goes wrong, and also interpret more biophysically what the fluorescence and OD numbers that we measure are actually telling us. So without further ado, let's actually look at a little bit of data. We've put some data up that you can play with if you want, with examples of analysis. If you go to GitHub, to this iGEM measurement tools organization, there's a fluorescent tutorials that has both plate reader and flow cytometry examples, and all the data that I'm going to show is up there, and all the analyses you can rerun yourself using MATLAB or Octave. So one thing that we want to do whenever we're working with plate reader data is to sanity check that data, the values that we're getting for our positive and negative controls, to make sure that they're falling into a reasonable range of the number of proteins per cell. We want to first check and make sure that these numbers are within the linear range of our calibrant, so that nice long diagonal slope. If your negative control might be right down near the bottom of your sensitivity, your positive control should not be saturated. If your positive control is saturated, you need to change your instrument settings. Since the calibrants let us estimate equivalent number of proteins per cell, we can compare what we're getting for the positive control to the number of proteins that's reasonable for a cell to produce. So in an E. coli, the total number of proteins in a cell is order of 2 times 10 to the 6th. These are numbers here I got from BioNumbers, the BioNumbers site linked below. Yeast has about 10-fold more. A human cell has 1,000-fold more than an E. coli. But this gives us a sort of starting point. If we have a strong promoter, we should be able to get a small percentage of that total. So with a very strong promoter, we could guess that we ought to be able to hit a number of at least 10 to the 4th molecules in an E. coli. And here over on the right, you can see I've put that 10 to the 4th as the upper red line. And this data that we're showing had about 3 times 10 to the 4th as the value that was coming out of the positive control. So that tells us, yep, our positive control is showing we can strongly express fluorescent protein in these cells. We don't have a basic turning things on at all problem. If you're not hitting a number like that, you should be worried that either your fluorescence isn't working right or that you just don't have the ability to express things strongly in the cells at all, which can be a big limiter for working. Your negative control, on the other hand, should be much smaller than your positive. In this case, I set the threshold to be about 10-fold smaller than your positive. And we can see that this is, in fact, under this 1 times 10 to the 3. That's right down towards the minimal possible to measure level in these cells. It happens. The reason you want to take a look at these is because if these are not true, it indicates something's going wrong with your protocol and your experimental data is probably going to be invalid. If your negative control, if your wild type cells are showing up as 10 to the 4th, fluorescent molecules per cell, something's wrong. Maybe your calibration's all wrong. Maybe you've got something, your media's totally dominating all your measurements. One way or another, you're getting a number that's not biologically reasonable for those wild type cells, and you need to be aware of it and use that to debug. Similarly, if you're not getting strong fluorescence out of your positive cells, maybe you've just done something wrong that's making all your cells terribly unhappy. Maybe your colonies aren't growing well. Maybe you've managed to deselect something. Maybe you're just not in the right cell line. One way or another, look at the controls, and they're going to help tell you whether the experiment is worth doing or not. The final example I'll show here is taking a look at some of the experimental data that was being calibrated and controlled by those controls. This is some interlab data from 2018. The important thing that I want you to get out of this is that we're being able to look at this relative to reasonable biological numbers here. Over on the right, we can see that all of our colonies have grown pretty well. They're all, all except J23104 are at 10 to the 8th cells or more. And J23104 is not too far behind, but they're all far, far above the minimum amount that we can measure in terms of count of cells. So there's lots of cells in these wells, and we can be sure of it because we're very far from that bottom red line. Over in the fluorescence, we were expecting to see a wide range of fluorescence, and indeed we do, even down to some that can't really be distinguished from autofluorescence. And our range of fluorescence that we got covered the whole range except for sort of a medium-low level. That would be a nice thing to try for some time in the future. Not much more to get out of it, just wanted to show an example of how we can see this and how we can interpret this and see that we're getting all the way from a lot of the very close to that 2 times 10 to the 6th total all the way down to almost nothing. The numbers make sense and are something that we can relate to models and how we've been thinking about this in a theoretical principle. So to summarize, once we've got calibrated units, we're in a good position to convert them to estimates of cell and molecule count. We want to be able to analyze this in terms of geometric statistics because that's what happens with genes. Our low values may be indistinguishable from the background, and we should just accept and report that rather than trying to do something funky with the fact that things may get close to or below 0. once we've done all this, we can interpret our data in relationship to realistic biological values using positive and negative controls to detect process failures and relating our experimental values to our biological intuitions. And with that, I'll pause to see if there are any last questions before we wrap up the seminar. All right, seeing no further questions, I'll wrap the seminar. Next week, we're going to have a follow-on where we specifically discuss flow cytometry. And if you have any questions about this or about that as it goes, please feel free to email the measurement committee or to come to the office hours that the measurement committee is running once every 2 weeks. Thank you very much.